we completed the initiation yesterday, where we saw that the RNA polymerase holoenzyme loosely binds with the -35 region and then further binds strongly with the -10 region, resulting in the opening of the DNA double helix for a length of around 17 nucleotides. And then the RNA polymerase, after a process of abortive initiation, finally succeeds in properly aligning itself on the template strand and then proceeds with the elongation phase. The moment the RNA polymerase proceeds with the elongation phase, the sigma factor of the RNA polymerase opens up, that is the RNA polymerase escapes the promoter, and now the core enzyme transcribes the coding region of the DNA. So with this, we move on to elongation. And at any point, this is a beautiful picture showing that at any point, if you look at the bubble, transcription bubble, so it's around, this bubble is roughly around, as you can see, 40. It's roughly around at given time, the RNA polymerase contains around 35 to 40 base pairs of the DNA nucleotides. And the DNA in the transcription bubble, that is the DNA is open for around, as I said, 18 to 25 base pairs. And the RNA remains, the portion of the RNA that remains twisted with the DNA is about 8 to 9 nucleotides, after which the remaining of the RNA hangs out from the DNA. So temporarily about 8 to 9 nucleotides of RNA remains base paired with DNA in a temporary RNA-DNA hybrid. And as the RNA proceeds forwards, as you can see, the RNA will now move forward. So as the RNA moves forwards, the DNA untwists at this end and again reanneals behind. So this is how the process continues. So as the RNA polymerase moves, the DNA untwists in front and retwists, I mean to say, goes back to its double stranded shape behind. So at any point, we have a transcription bubble of around 18 to 25 base pairs open. So this is how we can see the template strand, the holoenzyme binds, there's a promoter escape, and then RNA starts synthesizing, a polymerase starts synthesizing the RNA transcript. Now, coming to the part that I had told you that in the transcription, the polymerase recognizes the promoter, starts coding from the coding region of the DNA, and goes on transcribing and finally reaches the terminator, where it also transcribes the information of the terminator. So if we look into the terminator region of prokaryotes, we will observe that basically there are two categories of termination that occur in prokaryotes. So the first category is the Rho. Rho is a protein. Rho independent termination, or the Rho independent terminators, which are known as type 1 terminators. And there's a second category which is known as the Rho dependent terminators, or the type 2 terminators. For either of the terminators, if we look into the sequence of the DNA present here in the terminator region, we will see that there are subtle differences in the sequences of the DNA present in the terminator region. For Rho independent, when the Rho protein is not being used, so when termination is being done in that manner, you see that the DNA in the terminator region has a two-fold symmetry. A two-fold symmetry means that if you read the sequence AG, CC, CCCGC. And if you read it as T, so this is complementary, TCGGGCG. So this part of the DNA sequences is complementary. So this part of the DNA sequences is complementary to this part of the DNA sequences. So when they are transcribed into RNA, you see the sequence AGCCCGC. So we did it in this direction, and you see the sequence UC, and we did it in this direction, UCGGGCG. Since we have uracil instead of thymine, and this is a single-stranded molecule. So this molecule hangs, and then these two sequences tend to form a hairpin fold like this one. Since they are single-stranded, they tend to come together. They tend to attract each other, and they then form this pairing. So this portion was unique. So this loops out. So we find a hairpin looping of the RNA molecule in case when the DNA reaches the terminator region for row independent terminators. So what happens? And another thing to be noted here is also that apart from this two-fold symmetry found in the terminator region, you can see there is a poly T site. There's a number of T and A. Okay. So the template strand will be, this will be the template strand because this is 3', this is 5'. The RNA is copied. So you can see 3', 5'. So in the 3' we had A. So when we have a complementary strand of the template one, we have a poly U string. So in case of row independent terminators, two things are to be noted. The first thing is that there is a two-fold symmetry which is copied into the RNA, for which that portion of the single-stranded RNA can pair with each other. And then we have a poly U site. So this is T instead of T. This is the coding strand. So instead of T, we'll be having U in the RNA. So we have a poly U site. So these two factors are essential for row independent termination. Now what happens? Now let's see another picture. Let's look into the left side picture first. So you see, we have this two-fold symmetry. The RNA is coming out, and you can see that in the two-fold region, the RNA has paired. And now the remaining part of the RNA, the poly U part of the RNA is being synthesized. Now, since we are having a heavier region here, since we are having the RNA folding into a hairpin loop structure, the hairpin loop structure causes the RNA polymerase to slow and pause its catalytic synthesis of RNA. And after this hairpin, we are having this stream of U nucleotides, which destabilizes the pairing of the new RNA chain with the DNA template strand. And as a result, the RNA polymerase dissociates from the template, the chain dissociates from the template, and transcription is brought into a halt. This particular picture is from Snustad and Simmons, and this is a picture from Broca Genetics. The similar picture, we have that structure inverted repeat sequence as a result of which a hairpin loop is formed, and then this is the poly U rich region of the DNA, and as the polymerase reaches there, it is paused, its catalytic activity is paused, and ultimately the reaction here, I mean to say that the interaction between the polymerase and the RNA DNA hybrid is lost or destabilized, and the polymerase opens up, also the RNA molecule opens up. So this is about the type 1 terminators or the row independent terminators.
Ma'am.
Okay. So we completed that part where we studied the how RNA, how row independent terminators act. So there was no involvement of the row protein. Now coming to row dependent terminators. So row dependent terminators, when we are talking about, you can understand that there is a protein which is known as row protein, which binds to the C-rich terminator sequence. See, let's have a look at this. This is the terminator region. First, understand the picture. Let's understand the figure first and look at the positions. So this is the terminator region for mRNAs which are following the row dependent termination. And we have a small region upstream of the terminator region. So if this is the terminator region, so this is upstream of the terminator region. And this region is a C-rich region. So the sequences here are mainly cytosine sequences. And this site is known as row utilization site or the RUT site. So when in case of row dependent terminators, the polymerase goes on transcribing the RNA, mRNA, and it also transcribes this row recognition site into the mRNA. Once this site has been transcribed into the RNA, now row is a protein which consists of 6 subunits. Now these 6 identical subunits are then loaded into the mRNA and they start moving towards the polymerase. So what happens is that row actually, what is the function of this RUT site? The function of this RUT site is that row recognizes the RUT site, and you can see the subunits is the picture is shown in form of subunits only. The 6 subunits bind here and they start moving towards the polymerase. As they start moving towards the polymerase, as a result of which, now by the time when they are moving towards the polymerase, you can see the polymerase has completed transcribing the terminator part also. So as the row moves towards the polymerase, a stem loop is formed, and the RNA polymerase pauses due to its interaction with the stem loop structure. The row protein opens up the complex and then separates the RNA-DNA hybrid. So once more, the row binds to the RUT site, and the row then moves towards the polymerase, as a result of which a stem loop is formed. Because of a formation of a stem loop, the RNA polymerase pauses. Row then opens up the stem loop and catches the polymerase, and then it really catches up the polymerase, and its helicase is basically, it functions as a helicase. Its helicase unwinds the helix between the RNA and the DNA template strand using ATP hydrolysis. And the new strand is then released from the double helix. The RNA polymerase and the row proteins dissociate from the double helix. So you can see this is a comparison of the two pictures. This is a row independent termination where we had, as I said, we had this two-fold symmetry and the formation of the stem loop, and then we had a poly-U stretch. So this is how the RNA opens up. In this case, we have a C-rich region, as I said, the RUT site. The row protein binds to the RUT site. The row protein starts moving towards the polymerase, as a result of which a stem loop is formed. It's not shown here. As a result of which the stem loop is formed, row opens up the stem loop, and then finally, it being a helicase, unwinds the DNA-RNA hybrid. The polymerase is released, and finally, the row protein is also released, and the RNA molecule is also released. So this is how termination of two categories, one row independent and the other row dependent, is brought about in prokaryotes, in case of prokaryotes. So what we saw is that we saw the termination process of prokaryotes. Now, if I look into the replication cycle, so this is a beautiful picture of replication cycle. The book is from Pollard, Cell. If you are interested in reading the book, ask me, I can send the PDF. So you have this promoter element where the RNA polymerase sits and then binds to the promoter elements and escapes the promoter. As long as the RNA polymerase does not escape the promoter, we call it under initiation. Once the RNA polymerase escapes the promoter, it starts transcribing the DNA, and this portion falls under elongation. And when the polymerase reaches the terminator region, we saw that once it reaches the terminator region, depending on whether the terminator region has a two-fold symmetry or whether the terminator region has a RAT sequence, there are two kinds of termination. So the polymerase gets terminated and the polymerase is released. It can now enter a fresh cycle of transcription once the sigma factor joins to it again. Because if you remember, this is the core enzyme that is released. The core enzyme cannot recognize promoters. Once the core enzyme is released, it needs to be joined by the sigma factor to form the RNA polymerase holoenzyme, which will now again recognize the same promoter, can recognize the same promoter. If a particular gene needs to be transcribed very fast, we need a certain amount of protein very fast, then the moment the polymerase opens up from the terminator region, it can be joined by the sigma factor, and it can again sit in the promoter element of the same gene and again start transcribing that same mRNA. So this is how the transcription cycle goes on. And this one big difference is between the prokaryotes and the eukaryotes. If you look into this picture is particular from Russell I genetics. See, this is the bacteria. This is a prokaryotic cell, as you can see. And as I had said, that in case of prokaryotes, the RNA hangs. And you can see the ribosome has immediately been uploaded on the RNA and is now decoding the RNA to produce the polypeptides. So in case of prokaryotes, as there is no nuclear membrane, translation can begin. This is very important. Where is my cursor? Translation can begin while transcription continues. And this is coupling of transcription and translation. This phenomena is known as coupling of transcription and translation, which is found exclusively in prokaryotes because prokaryotes lack the nuclear membrane. In case of eukaryotes, we have a more complex structure. We have a definite nuclear membrane, and because of this presence of the definite nuclear membrane, you can see the polymerase is synthesizing and the RNA has come out, and you can already see the 5' cap has been added in the 5' end. And then once the RNA is released from the eukaryotes, you can see we are having the primary transcript and the poly A tail, and then we have the final mRNA where the introns are removed, and this mRNA is then moving into the cytoplasm. And this mRNA is then being recognized by the translational machinery to decode into a polypeptide. So in case of eukaryotes, only when the mRNA is transported to the cytoplasm can translation occurs. So there's a big difference between the bacterial setup where we see the case of coupling of transcription and translation, and in the eukaryotic setup where we are seeing that once the mRNA is being transported after the addition of a 5' cap and a 3' poly A tail and the removal of introns, then only in the cytoplasm can the translation start by the translation machinery. So, and we'll do eukaryotic initiation tomorrow. So I'll stop the class.